Today I have the pleasure of introducing Dr. Jack Hoops for this talk. First off, thank you for joining Dr. Hoops. A little bit of Dr. Hoops' background. Dr. Hoops is a veterinarian with specialty training in veterinary pathology and academic graduate training in radiation biology, radiation oncology, and experimental cancer therapeutics. Dr. Hoops has his postdoctoral training in experimental radiation and hypothermia cancer therapeutics. Dr. Hoops has over 25 years experience in the development and use of animal models for many types of translational biology, imaging, and therapy, including MRI, CT, ultrasound, PET, fluoroscopy, and fluorescent imaging for large and small animal models. Dr. Hoops is continually funded by NIH and corporate ventures for more than 25 years. Dr. Hoops is the director of the Dartmouth Center for Comparative Medicine and the Dartmouth Norris Cotton Cancer Center, based irradiation and small animal irradiation shared resource group. Dr. Hoops' current lab consists of 4 PhD students and 4 undergrads. Dr. Hoops has mentored more than 25 grad students and 30 surgery residents in the past 25 years. So without further ado, please welcome Dr. Jack Hoops, and I'll turn it over to you, Dr. Hoops.
Yeah, Jason, can you hear me okay?
Yep, I can hear you.
Okay, good. Yeah, so thank you very much for that introduction. Yeah, what I'm going to talk about today, I think it's just about 10 minutes, is really our movement at Dartmouth from cesium experimental irradiation to the XRAD 320 to X-ray irradiation. And so Dartmouth has had, I think we wrote an S10 grant about, maybe 30 years ago when we moved to the new hospital facility. We have a 12,000 curie cesium irradiator that has been a workhorse and has really basically irradiated all cells and mice at Dartmouth over these past 30 years. Of course, with the recent movement of the federal government to sort of replace all the cesium irradiators and so-called cesium irradiator removal program, we entered into that program and made a contract with the government, and which is run by the Pacific Northwest National Lab, the PNNL lab. And they, so we made an agreement with them to get rid of our cesium and replace it with X radiation. So we've been in the process of that for the last 18 months. We currently still have the cesium irradiator, but all of the investigators have moved to the XRAD. So, I'll have to say, and many of you know who are interested in this, is that, it wasn't necessarily an easy move for many people, because there's this ongoing controversy about whether the relative biological effectiveness of one, say, rad or one gray of cesium has the same biological effect as one rad or one gray of X-rays. And we usually say that they're comparable, and most people believe they are comparable. Clearly, the energies are slightly different. Our new XRAD instrument is 320 kV, and the cesium is 662. So there is a little bit of difference. So, but basically for mice, at least the things we're doing, and for cells, we sort of have believed that it was similar. Like I said, we've converted over now, and I'm fortunate because I have a radiation oncology department that I work with, obviously part of the cancer center, a lot of radiation physicists who have helped us. So we've done some reasonably extensive assessment as part of our agreement with PNNL to look at the cell survival efficacy of treating with cesium and X-rays, at the same dose. And I'll show just a couple of quick slides here. I'm going to do my screen share, and it is this one. Let's see, share. Yeah, so if you can see this, 2 different cell lines, B16, F10. I'm going to show that one first. And if you can see these lines, cesium, X-ray, LINAC, different doses, percent survival. These are colony forming assays. All of you know about that. So it looks like they lay really pretty much on top of each other at these different doses. So that was very encouraging. And then the other cell line that we used was a U87 cell line, a more